If we want to fully understand how psychoactive drugs work, we need to do more than just understand how drugs affect behavior. We also need to understand the mechanism of action and how drugs affect the nervous system. To do this, we need the techniques of neuropharmacology. These techniques can then be used to complement what we've used from behavioral pharmacology to get a fuller picture of how drugs affect the nervous system and why that leads to their particular behavioral effects. So next, we're going to review some of the most commonly used techniques in neuropharmacology to help you understand how we learn the mechanism of action of drugs, as well as how various neurotransmitter systems affect different aspects of behavior. We can divide the neuropharmacology techniques into techniques that allow us to manipulate the brain and techniques that allow us to measure the brain. And we can do these across species in rodents, in monkeys, and in humans. A stereotaxic apparatus is used in rodents and monkeys and humans to precisely target neural structures. We have maps of the rodent brain, the monkey brain, and the human brain, which means we can assign different regions of the brain different coordinates. And then the advantage of using an apparatus like this is that we can fix the animal's head and keep it entirely still. This allows us to then drill a hole in the skull and implant something at a specific location. And we can do this in 3D by using 3 different coordinates, which we call the medial lateral plane, the anterior posterior plane, and the dorsal ventral plane. Each structure we're interested in has a particular location, and we can look up the coordinates for that region. Then we can drill a hole in the skull and implant something precisely at that location. This allows us to do a number of different things. So the stereotaxic frame is really very central to neuropharmacology research. For example, we can implant a cannula, which would allow us to infuse a drug directly into the brain. This means we could test the effects of that drug in various regions of the brain rather than having to give it to the whole animal systemically. But all of these techniques are possible because of the stereotaxic frame and the advantage it has in letting us be very precise in targeting neural structures. Once we've targeted a certain brain structure, we can do a number of things. One example is that we can actually collect fluid from the brain and then analyze what's in that fluid. This is really useful in assessing the amount of neurotransmitter that is located in a particular brain region at a certain time. And this technique is called microdialysis. You can see an example of this diagrammed here. An animal has a cannula implanted in a particular brain region. You can see that's zoomed in on here. And then tubing is inserted through the cannula. And the tubing is semi-permeable at the tip. And so this works through the concept of dialysis, much like you may have heard of kidney dialysis, that fluid is moved through this tubing, and because it's semi-permeable, anything in the surrounding area will also flow across the membrane, and that means that the dialysate that comes out of this tube will contain some of what was in the extracellular fluid here in the brain. And so then we can pump this all out and collect it, and we can do this while an animal is performing a particular behavior. We can then look at what was in the dialysate and say, during XYZ behavior, say performance of a certain maze task or an operant behavior, we saw glutamate being released in that the glutamate levels increased, for example. So this technique has been really foundational in understanding how different neurotransmitters are released during certain behaviors. And we could also give a drug and then examine how change in a particular neurotransmitter is occurring when that drug is given. So an example of this would be in the dopamine system, where microdialysis has been very foundational in understanding that all drugs of abuse cause increases in dopamine release in certain areas of the brain. Another thing we can do with stereotaxic techniques is implant electrodes in the brain. And so you see this diagrammed here for a rat, where the electrode here and the wires coming down have been implanted deep into the brain, and then they're cemented in place, usually with a dental acrylic. And this allows us to do two things. One, we could stimulate the brain, and that allows us to see what is stimulating different neurons due to behavior, or to the effects of different drugs on behavior. And it also allows us to record from the brain. So this is a good example where we can both manipulate the brain as well as record and gather information from the brain using techniques with electrodes. And that's very useful. For example, we can examine neural firing rates in particular brain regions as animals perform behaviors and in response to administration of various drugs. It's important to always remember that so many of these techniques that we can do in rodents can also be done in non-human primates and in humans. So this is an example of electrical brain stimulation and recording in a human and what that might look like. Now, we don't do this very often because brain surgery is a serious business, but some people need brain surgery to correct certain conditions, and that means that occasionally we do have the opportunity to study the electrical firing of the human brain. And a number of different disorders are now being treated with brain stimulation, and so electrodes are implanted into brain regions to deliver current that can normalize some behaviors. This has been successful, for example, in treating Parkinson's disease, where stimulation of the brain can restore some of the normal movements in those patients. More recently, in the 21st century, we've seen the invention of techniques that use viruses to control neural activity. You may have heard of a very common technique these days called optogenetics, which uses light or optical stimulation and genetic infection of neurons via viruses to control neurons. With optogenetics, we can put an animal in the stereotaxic frame and inject a virus in a specific location. Viruses will infect neurons, and we can engineer those viruses to carry genes that we're interested in. So the basic optogenetic setup is to inject a virus that carries a gene for something called channel rhodopsin. And channel rhodopsin is a light-sensitive protein. So when the virus infects the neurons in the brain, those neurons will begin to make this protein channel rhodopsin, and it gets trafficked to the cell membrane and becomes functional. But channel rhodopsin is an interesting channel in that it doesn't respond to voltage or ligands like we've talked about before. Instead, it responds to light. And of course, usually there's no light in the brain. And so this protein will simply remain inert and not do anything to neurons until we shine a light. And that gives us very specific control over neuronal firing. So we can implant a cannula here that will allow us to insert an optic fiber into the same area where we injected the virus, and then we can shine light in very brief pulses. Channel rhodopsin is sensitive to blue light, and so any neurons that have been targeted and are expressing channel rhodopsin will respond with action potentials when blue light is shown. That's because the blue light causes the channel to open and positive ions flow into the cell. So we can mimic different types of neural firing and stimulation patterns in different brain regions using optogenetics. And this allows us to ask questions about how different brain regions control behavior. In addition to channel rhodopsin, which passes positive ions such as sodium into the cell to cause depolarization, we can insert other proteins, such as chloride passing channels, which will cause inhibition. So we can also turn brain regions off using optogenetics. Now, you probably won't be surprised to hear that we can't always do stereotaxic techniques. We can't always implant something in the brain. That's especially true when we're doing research in humans. It's very rare that we get the opportunity to directly measure from the human brain, for example, with electrical recordings. So imaging techniques are very important, particularly in human studies of neuropharmacology. And there are a number of different techniques we can use that have different strengths and weaknesses, and that we use in different experimental situations. CT scans, which use X-rays, and MRI, which uses magnetic resonance imaging, are both really useful for looking at the structure of the brain. These techniques can give us detailed images of the structure of the brain and where different types of tissues are. We can see gray matter, which are the neurons of the brain, and white matter, which is the myelin or glia. And we can resolve those structures with a pretty high degree of detail, particularly using MRI. So this is important if we want to look at the volume of a structure and maybe how it's changed in certain individuals, and of course for a variety of health-related applications. In neuropharmacology, another particularly useful technique is PET scanning, or positron emission tomography. This technique allows us to image radioligand binding in the brain, and so we can actually administer individuals, and you see here this person is being given an injection. We can administer them different pharmacological compounds that will bind to things we're interested in the brain. So this might be various neurotransmitter receptors, for example. We can put in a ligand that would bind to a receptor that we're interested in, and because it's tagged, we can then image with PET scanning where that ligand binds. So we can find out where neurotransmitter receptors are in the brain, and we can also learn about neurotransmission or neurochemical signaling at those receptors by looking at changes in that signal. So if we see binding to in a particular area of a ligand we're interested in, and then we ask the participant to do a certain behavior, or we give them a different drug, and we see changes between the two conditions in the binding of our ligand, it will tell us that either the drug or the endogenous neurotransmitter for that receptor has been released and is competing for binding to that site that we're interested in. In addition to manipulating and measuring inside of the brain, we can also measure proteins and DNA levels by taking brain tissue out of the body and looking at it in vitro, or in a dish. One example is immunohistochemistry, which uses antibodies to look for proteins of interest. We can make antibodies to all different kinds of proteins, and then apply those antibodies to tissue that we've taken from the brain, and they'll bind to the proteins that we're trying to find. We can then use what's called a secondary antibody, which these days is commonly attached to a fluorescent tag. This means when we visualize the tissue under a microscope, we can see fluorescence where our proteins of interest are located. So for example, here we can see labeling of the dopamine type 3 receptor in neurons of the rat striatum. And we can tell that dopamine type 3 receptor is actually only localized to some of the neurons in this region. The green labeling is for the protein that makes the dopamine receptor, and the blue labeling is for neurons. So you can see there's only overlap between some of those neurons.